Okay, let me start over again. This is Dr. Grenier, and I am doing a Bio 151 lecture. All right, I've made sure I turn the talk button on, so let's go through this again. The objective of this class, you know what? I'm going to start over, and I'm going to delete this session because I think it's going to confuse people. So hang tight if you're listening and join the other session. All right, everyone. I had the talk off, so hopefully you're hearing this now. Here is our lecture over cells. This is Bio 151 with Dr. Elizabeth Grenier. This week, week 2 of our class, it's all about cells and tissues. In your textbook, it is lacking any chapters or any specifics over cells and tissues and the pathologies associated with that. So you'll notice that I had to put together some information related to what are cells, what is tissue, and then what are pathologies that would be found at the cellular level or due to a cellular issue, or at a tissue level and due to a tissue issue. Okay, so that's our goal this week. Your worksheet for this week is over cystic fibrosis, and part of the reason why I chose something specific to cystic fibrosis is there's a problem in one gene for one protein, and that protein plays a role in every cell potentially in your body because it makes a faulty chloride channel. And if chloride cannot move into and out of the cell appropriately, whole body-wise, that's a problem. And so that's why I chose it. But other pathologies do exist that occur at the cell and at the tissue level, so we'll try to go through a few of those in lecture. Okay. If I don't get to tissues today, we will try to play catch up tomorrow. I'll try to do a mini session. Worst case scenario, there'll be a mini session over tissues on Friday. Okay. So let's review the cell. All right, cells. They come in a variety of sizes and shapes. They are typically microscopic, but look at an ostrich egg. That's one of the largest cells in the world, so they can be very big. In humans, because this is a human class, almost all of our cells are microscopic. And they are going to have different shapes based upon their function and in some cases their structure. The cells are typically going to contain some type of boundary, a membrane, and then within the membrane, the cell proper, it's going to have some cytoplasm, some water, some combination of dissolved ions, proteins, other molecules in a watery background substance. In addition to cytoplasm proper, there will be more advanced kind of structures, specialized structures, and those are called our organelles. Okay, and there's usually some proteins that form the cytoskeleton and proteins that perform enzyme functions, proteins that are signaling molecules for organelles to be able to talk to each other. The plasma membrane is the outer layer. And so if I highlight, so it's kind of at the bottom here. Sorry. All right, and the plasma membrane is going to be composed of a phospholipid bilayer, so 2 phospholipid layers, the fat-containing part towards the center. And in that fat-containing part, kind of on both membranes reaching towards each other, we make a hydrophobic or an unfriendly part of the membrane for water. And in doing that, we have then a barrier that has a tendency to keep water in or water out and helps us create an intracellular environment versus an extracellular environment. Now our membrane is not completely impermeable, so it will have potential for some molecules, if they meet certain criteria, to be able to move through it pretty easily. And anything that doesn't move through because of the hydrophobic feature of the lipid component will potentially have to have some type of gate or channel or pore made that can be open all the time, open part of the time, or open under certain circumstances. So that's your plasma membrane. Okay. The nucleus or the nucleolus is going to be typically the control center of the cell, and the nucleus is going to tend to be where the DNA of that cell is contained. and the DNA is our genetic code. As a human, you have 46 chromosomes. You have 23 pairs of genes on these chromosomes. And so you normally have 2 copies of everything. So for some of our cell issues, you not only have a defunct or a problematic single gene, you normally have a problem or defunct coding of 2 genes. And it just so happens to be the exact same gene on each of your pairs of chromosomes. All right. Now, the nucleus does have a membrane, so there's going to be some more, phospholipid bilayer, but it's a little bit less restrictive, so there are bigger sized openings and pores for things to be able to come into the nucleus versus out of the nucleus. And the nucleus is going to be like spaghetti and meatballs. So the meatballs are going to be the nucleolus, this dense area with RNA and some additional molecules. And then the DNA is actually like spaghetti noodles. They're just kind of long, stringy material all around the nucleolus of the nucleus itself. Okay? So the mitochondria is sometimes nicknamed the powerhouse of the cell. And it is a 2 membranes, so it has an inner membrane and it has an outer membrane. It's 2 membranes, all right, so 2 phospholipid membranes that make these kind of sac-like jelly beans or peanut-looking things in the cell. And it's thought that mitochondria might at one point have been bacteria or viruses that a cell ate but didn't completely kill or remove because it had this machinery, this setup of genes that was more efficient at making energy. And the goal of the mitochondria is to make energy. And in doing so, it helps break down carbon molecules, breaks apart carbon-carbon bonds. That energy is then harnessed to make a phosphate-phosphate bond in the molecule adenosine triphosphate, and oxygen helps with that process by dealing with some electrons. So a lot of chemistry stuff goes on at the mitochondria. The mitochondria is one of the only organelles that actually has DNA. And what's cool about the mitochondria, if you think this is cool, is you get all of your mitochondria from your mother. So the egg cell in humans has mitochondria. The sperm does not. So everyone's mitochondria, and so all of your genes in your mitochondria come from your mother. And so they have done some research to trace back with mitochondrial DNA testing how many potential females gave rise to the entire human population. So there's some cool stuff related to mitochondria and DNA. Should your mitochondria DNA have damage or problems, that can lead to a potential cell issue. Because if there is a gene encoded in the mitochondria that's problematic, that could lead to problems making energy. Problems with energy means the cell might not work properly, problems at tissue, organ, and human body level. Okay. Endoplasmic reticulum usually have either smooth or rough. Smooth means there's no presence of ribosomes. Rough means ribosomes are present. So endoplasmic reticulum in many ways is kind of a way to have these sacs, these canals within the system that you can sequester different products. Normally when you have rough ER, you're making proteins. And when the proteins are being coded and translated into a single chain of amino acid bonded molecules, it then enters the rest of the endoplasmic reticulum, the sac, and continues to be further processed into a functional protein. That might mean a few snips and cuts, trims up some amino acids, so there's some post-translational modifications that occur to the protein in the protein. Smooth ER is sometimes associated with an area to make phospholipids and other fat-type molecules. Again, fat doesn't like to be in water, so it wouldn't want to be in the cytoplasm. So smooth ER provides a little spot within the cell where you could potentially do fat-based chemistry, make fat molecules or break down fat molecules or make phospholipids, make cholesterol, make fat hormones, and you can kind of do it in a sequestered little part of the endoplasmic reticulum. Ribosomes are one of the 3 types of RNA. So it's usually a combination of some protein and RNA, typically ribosomal RNA. And it's a docking station for messenger RNA to dock and transfer RNA to come together and encode and translate into a protein. So it's a part of protein making. The Golgi body is sometimes known as your FedEx or your packaging center. It's also a flattened sac of like endoplasmic reticulum. It kind of has that membranous layer that makes this little sac. But what happens in the Golgi apparatus is the sacs are kind of quarantined and certain molecules are brought together, and then they peel off into little vesicles that can either hang out as entities by themselves in the cytoplasm, or can head towards the membrane of the cell so they can be released to the extracellular environment. Right? And a lot of times proteins make really nasty enzymes, like digestive enzymes or enzymes that can, if let loose, could eat the cell from the inside out. That could be toxic. So the Golgi helps us kind of quarantine and keep those molecules from being activated, from being available and potentially damaging to the cell itself. Because cells have to make nasty little products sometimes. Okay? Peroxisomes and lysosomes, these are some of the fairly newer defined and found organelles. Lysosomes we tend to call our trash man. They are full of digestive enzymes, and when the cell eats or brings in molecules from the outside world, in many cases the vesicle, that endocytosis, merges with the lysosome, and the digestive enzymes in the lysosome break it apart into useful smaller pieces. And the peroxisome can do that somewhat, but it can do it with oxidative type molecules. So peroxide, hydrogen ions or anions and hydroxide ions and things like that. All right? And the cytoskeleton, of course, is part of the cytoplasm. It's usually made of some different proteins known as microtubules and microfilaments, and these give the cell potentially structure, length, roadways, transportation routes for things to move between the different organelles to the membrane, or the membrane to the nucleus, and then can play a role in moving the cell itself. So with muscles, it's a lot of the organization of the cytoskeleton proteins into specified groups, and potentially a more robust organelle known as the myofibril that then lead the muscle to be able to contract and shorten or elongate and relax. The centrosomes and centrioles are going to be organelles that tend to play a role in cell reproduction. So we are not usually going to see centrosomes and centrioles in terminal differentiated cells, muscles, neurons, red blood cells, because those cells, they don't ever reproduce. They don't go through mitosis. They just disappear. They get killed off. They get destroyed and recycled. But in a cell like your connective tissue cells, your white blood cells, your skin cells, any of your epithelial cells, they might have centrosomes and centrioles because these cells do need to undergo mitosis to replenish and replace themselves. And so they'll have this organelle. This list is not all-encompassing. Again, I mentioned in the muscle, they have unique organelles, one being the contractile unit, the myofibril. They have sarcoplasmic reticulum, so it's basically endoplasmic reticulum that sequesters calcium. And then you have other cells like red blood cells that have virtually little to no organelles. So red blood cells basically have some cytoskeleton and some enzymes. That's it. They have jettisoned away their nucleus. They've gotten rid of their mitochondria. Sperm, same thing. They've basically cut down to their nucleus and some enzymes. You know, so depending on what cell you're looking at, they may or may not have this list of organelles, and they may have additional or other organelles that are only found in maybe that cell, so we normally don't talk about it in a general cell talk. Okay? So just to kind of remember, the plasma membrane has to be a barrier for the cell. It's got to keep things out and keep things in, and it does that by playing with the water-loving versus water-hating components of molecules. So the base of the plasma membrane is 2 fatty acids bent towards the center and a water-friendly component, a phosphate, on the outside. And when you put this together, so all these little like feet-looking objects, and then interspace there's some little balls, that's cholesterol. All of that is fat molecules. And fat molecules do not want to be in water, so they tend to clump into this inner part of the membrane. And any water-friendly parts of the membrane are going to be towards the inside and the outside. Now, this membrane, sure, could things possibly flow between the balls of the phosphates, between the legs of the fatty acids, and then inside? Yes. But for a molecule to do that, it has to be very small, and it has to be something that is water-soluble, so it can be inside and outside the cell, and then it has to be able to very easily become fat-soluble, and that means flow into a fat-rich environment. Very few things do that. Oxygen, of course, is a dissolved gas, and carbon dioxide, another dissolved gas, and carbon monoxide could do this. So this is why oxygen and carbon dioxide can flow freely from inside to outside of the cell. And they flow purely based on concentrations, based on diffusion levels. Okay? Most of our other molecules, they might have a charge, like an ion, so they are a positive or a negative charged molecule or atom. They are not going to be able to be in a fat-rich environment. So they have a problem getting into or out of the cell because they can't pass in a hydrophobic or a fat-rich environment. And so we get around that by putting proteins inside our membrane, and the proteins form circles, form holes, form pores that might be big enough and might be able to then allow bigger charged molecules to flow into and out of the cell. And sometimes with these proteins we put doors, so we only let them be open or closed in certain situations. Right? And so because of that unique quality, we are going to have a plasma membrane of our cells that controls entry, controls exit, and has an ability to keep things in and keep things out. Now, part of the way cells grow is they have to respond to their environment. So you'll notice that sometimes the molecules embedded in or as part of the membrane are going to have these extensions that extend into the intracellular or the extracellular environment. And these extensions come from, different pieces and parts of the protein itself, or sugars or carbohydrates that get attached. And what happens is it allows the cell to potentially interact with something out here in the extracellular or something in here in the intracellular environment. And so that's how a cell can communicate. So if you have a hormone or a protein, let's say you have growth hormone, okay? And growth hormone is really big, and it isn't fat soluble, so it can't just flow into the cell. But it can latch on and maybe bind to some of these outside reaching portions of proteins and glycoproteins. And in binding and interacting with those receptors, it could trigger things inside that then lead to the effects of growth hormone. And so that's what your plasma membrane can do. Okay? So getting things into and out of the cell can happen by usually one of two mechanisms. Passively, meaning there's no energy expended. The cell can let things come in or out of the cell based on the chemical gradient, the concentration gradient, or by the electrical gradient, by the electrical properties of things inside or outside of the cell. And there's a few different types of passive transport. The one most people are familiar with is known as diffusion. And diffusion means any really small molecule passes through a membrane without any assistance based upon the concentration and electrical properties inside versus outside of that cell and the amount of that molecule itself. So things that usually diffuse readily: carbon dioxide, carbon monoxide, and oxygen. Facilitated diffusion just means the molecule is going to move into or out of the cell, but it might need help because it might not be able to pass freely through the membrane. So it might have to have a protein to make a hole for it. It might have to have a protein to bind it and then bring it in. So it just needs a little bit more help. Even though it has a protein, there's still no energy expended. So the things can move in and out of the cell without any energy being, or ATP being utilized. All right. Passive transport in the specifics to water molecules gets its own name, and that's known as osmosis. And osmosis means water will move based upon the solute concentration. So based upon the amount of molecules that are ions, proteins, carbohydrates, amino acids, nucleic acids, any small molecule that's dissolved in the extracellular fluid or the intracellular fluid. Okay. Anytime you have to do active transport, energy is involved. And normally what that means is you're trying to move a molecule from the area where there's little of that molecule, and you're trying to put it where there's a high amount of that molecule. So you're moving something against its concentration gradient, and you can be moving something against its electrochemical gradient. Okay. The other type of transport is usually vascular transport, and tends to be used when you have something really, really big that you need to bring in, or like those digestive, sequestered enzymes that you want to release out. So this usually involves vesicles that are going to be bound and brought to the membrane and releasing its contents, therefore joining the plasma membrane, or vice versa. It's a pinching off of part of the plasma membrane. Right? Now let's talk about some of the pathologies or diseases that can occur. So a pathology or a disease of the cell is defined as anything that can result in an inability of a cell to release or respond to its outside environment. Right? And so there are some diseases and conditions that are due to your inability to exocytose chemical messengers, proteins into the extracellular environment. And one example of that is botulism. So botulism, if I remember correctly, is sometimes also known as lockjaw. Okay? And in this problematic disease or condition, what you have is your vesicles cannot dock appropriately. So there's some proteins listed down there known as snare proteins and syntax and SNAP25 and somata blah blah blah. Okay, so those proteins don't interact correctly. And because they don't interact correctly, the vesicle never releases its contents. So because the vesicle never releases its contents, the acetylcholine, the protein in there, the molecule that's going to tell a muscle cell to contract, never occurs. So you end up in a situation where the person cannot contract their muscles, so they are in a locked or a fixed state of paralysis because they don't have the ability to release neurotransmitter. Okay, so that's a condition or a pathological disease because of a problem at the vesicle level and the cellular level. Okay? All right. Now, other problems sometimes occur because in order for cells to be able to carry signals, interact with the world, they have to have certain concentrations of certain molecules inside versus outside, and they tend to collect certain charged ions in or outside of the cell. And so in addition to making concentration gradients, they also make electrical gradients. And when cells do this, they have what's known as a resting membrane potential of some of the ions inside versus outside are a little different in both their concentration, their physical amount of ions present, as well as the electrical properties associated with those ions. And we use these kind of different concentrations of ions to help make cells like neurons and muscles be able to generate what's known as electrical impulses, also known as action potentials, and graded potentials, which are just little ripples that can occur across the membrane as ions enter and leave the cell, how that happens. Okay? And I don't want to go through a lot of details because that's anatomy and physiology and like weeks of that. So just know that there tends to be potassium high inside the cell and little potassium out. And there tends to be a lot of sodium outside the cell and a little sodium in. And when certain situations occur, sodium comes in and that messes and changes concentration and electrical properties of what's happening on that cell. And that can generate an action and or a graded potential. And then the cell works very hard to push sodium back outside and then reestablish its resting membrane potential. Okay? So anytime, I just mentioned sodium and potassium. Are they the only ions that a cell will try to keep in or keep out? Of course not. There are chloride ions. There are calcium ions, there are magnesium ions, there are lots of different types of sodium ions. There's some that are chemical gated, some are voltage gated, some are ligand gated, meaning they have to have a special protein. Some are co-transporters, some are anti-transporters. So very quickly this concept of ions are kept in or out of the cell, how they get in or out of the cell, the facilitating protein, the facilitating channel, might be one of many different types. And if you were to have a damage or a genetic messing mutation to one of those genes, you could end up with a plethora, as this slide shows you, of many different potential diseases because one ion channel is messed up. Okay, and so cystic fibrosis is an inherited condition where the channel, the protein that allows chloride ions, all right, to flow in and out of the cell is actually non-functional, and in some cases it's completely missing. So because chloride ions cannot be transported and follow sodium ions into and out of the cell, all right, you're going to have a problem then with water flow. And you're going to have a problem with the negative ion concentration gradient and some of the balancing of electrical signals because negative ion chlorine can't move into or out of the cell. Okay? And so the end result is because chloride can't follow sodium into and out of the cell, water doesn't follow sodium as effectively. And so when water doesn't follow sodium, you end up with potentially thickened secretions that are made when cells try to make mucus, make serous fluids, make secretions from their exocrine glands. And so the result, again, at a very simplistic level, is chloride protein that makes a hole for chloride is non-existent, non-functional. The whole body effect of that is in your lungs, in your respiratory tract. You have an abnormally thick mucus that could possibly clog and impair airways and prevent breathing or good air-gas exchange, and then that can frequently lead, in addition to the mucus is thick, it clogs, bacteria lives in there, and then you have higher rates of lung infections and other problematics to your lung. Right? And this was discovered in, I think, the 40s and 50s, and it was found in children. They had difficulty breathing, difficulty with digestion, again, because they couldn't make mucus in their GI tract. And it normally occurred in families, and what they found is it's a genetic disease. So both mom and dad each have a defunct gene. They donate defunct genes to child, so the child has no functional chloride channel, and so therefore develops, but develops with the inability to make proper mucus in the epithelial exocrine glands throughout their body. When we see the effects most pronounced in the lungs, but you can see issues in the digestive tract. Again, anywhere you have epithelial secretions such as mucus and fluids. All right. Now, we've learned a lot more about the disease, so it's not a death sentence in young anymore, but these people have to be very cognizant or aware of any exposure or any high exposure to respiratory illnesses, because their respiratory tract is just a mucus-rich region that's hard to clear, so bacteria can thrive there. It's kind of like in little kids, they're more prone to ear infections because they can't drain their inner ear, their middle ear, and so bacteria just lives and grows and grows and grows in there, and they have ear infection after ear infection. So they get tubes, they get a hole that releases that fluid. Well, this mucus, you can't clear it very well, so the lungs get problematic, and the lungs eventually can be an issue. Okay. Another issue is like cholera. So cholera is a bacteria, and if you read any literature, Love in the Time of Cholera, I guess, is this big book. So cholera is a bacterial infection, and I think there's books of like cholera outbreaks with war camps and cholera outbreaks in areas where they don't have good sanitation, they don't have good water. And what cholera is, it's the bacteria. You ingest it in, and it causes epithelial cells that have chloride channels, to leak more chloride. And as they leak more chloride into the interstitial space or the extracellular space, water follows. And so people with cholera basically have excessive, extreme diarrhea. And that extreme diarrhea is because chloride is being released into the lumen of their GI tract, and it's pulling water from osmotic gradients with it. And with that happening, the body will very quickly result in dehydration, severe dehydration. And severe dehydration ends up being severe loss of blood pressure. Loss of blood pressure then starts to be problematic for getting blood to the brain, getting blood to the major organs, and you start going into organ failure and death. All right, now, back in the early 1900s and before, we didn't have this thing called antibiotics. So getting cholera could be a death sentence, because you could die before your body started making antibodies against the bacteria. Okay? So those are just some of the examples, and again, most of those are related to the chloride channel. Doesn't always have to be something related to the chloride channel, but a variety of diseases and illnesses occur because of a gene issue, and a gene issue by a single cell, and that then leads to all of these diseases and problems. I'll just mention diabetes. Diabetes is a condition because insulin cell-making cells die. They get targeted for death, and your pancreas for type 1 diabetes no longer makes insulin. You don't have a cell making insulin. Now you have a whole body condition, type 1 diabetes. Okay? So that's just, again, a cellular disease that can then potentially cause problems for the whole body. All right. The other side of where cells can cause disease and problems and pathologies is when cells inappropriately go into cell cycle, or their genes controlling cell cycle get messed up. And so the cell ends up becoming over-responsive, so they begin going through cell cycle too fast, too quickly, and become cancerous. Or they can grow too large and cause problems because they become hyperplasia. So there's a bunch of issues that happen because cells get messed up in their cell cycle regulation. So there are genes within your DNA that control the cell's life cycle. They tell the cell when to grow. They tell the cell when to make copies of the DNA. They tell the cells when to go into mitosis. And for cells that have to have the ability to undergo and go through mitosis, this is a very controlled, very regulated process. And so when problems occur that lead the cell to inappropriately go into cell cycle, you can end up with the cells either dying and going into cell death, and that can be an issue if you lose, like in the pancreas, you lose all your beta islet cells that produce insulin. Or you can have the opposite, where the cells grow so fast, so quickly, that they start to make a tumor, and that tumor could be able to break away and move, or that tumor might lead to sloughing off of cells that individually leave and go and become part of the other part of the body. Right? So inappropriate cell cycle, inappropriate gene expression and regulation of this process of interphase and mitosis can potentially lead to cancer. Okay? So cancer is another illness, a pathology that can result because of abnormal growth, proliferation of any cell in your body. And when those cells form a mass, a group, they become what's known as a tumor. So a tumor is when the cell division and growth exceed the rate of cell death, and so excess cells begin to be found in an area and they enlarge into a mass or a group. Benign tumors are cells that are usually within epithelial tissue, and they have a capsule, and they're kind of separated. So they're kind of not necessarily into the bloodstream. They're kind of just a separate little entity, a little kind of like mole on your skin. Okay. When the tumor is malignant, the cells no longer respond to any of normal controls that would potentially arrest or stop their growth and proliferation. They are no longer confined to their originating area, and so they can spread to surrounding tissues. And that's usually a bad thing, because if they spread to tissues like the liver and the spleen, and then they can get into other areas of the body, the brain, the spinal cord, and they can start to have effects on those other organs. All right? And so how do cells become cancerous? Some of the genes involved that control growth, that control the cell to differentiate into its terminal, type, whether it's a muscle cell, a nervous cell, or a supporting cell, get screwed up. Something happens in the genes for controlling mitosis, cell division. They either get modified, they get mutated, they get permanently turned on, or they get permanently messed up. And in doing that, they then get turned into inappropriately regulated genes, and the cell then inappropriately becomes a growth factory and a cancerous cell. So some of the terms associated with cell pathologies in cancer are oncogenes. These are genes that when modified lead to increased risk of problems with cell cycle, cell differentiation, cell division, and cell control. All right, and they are going to be genes that, if modified, if damaged, if mutated, have the risk of making that cell be more likely to become cancerous. Okay, mutagens, or any kind of agents that can damage, alter, mutate, destroy DNA. So radiation is a mutagen. Radiation comes from the sun. And part of the reason why your skin is at high risk of becoming cancerous is because your skin is exposed constantly to radiation from the sun. And that radiation could damage cells. And if it starts damaging the cells of the skin, especially the genes related to cell cycle, you could then start to have a cancerous mass or cancerous cells growing in your skin. All right, carcinogens are cancer-causing agents. They can include mutagens. But we tend to think of carcinogens as like chemicals. So in our pesticides, we tend to put molecules that kill weeds, kill insects, and in doing that, if we ingest them, they can be cancer-causing agents to us. Cigarette smoke, the tar, the nicotine, some of that might be carcinogens to us. So sometimes the carcinogen is a chemical that we ingest that then can lead to cell death and problems and then cancer. Okay, so those are our cell pathogens, cell cell cycles. So what I want you to take away is cells are the smallest unit of life. Cells are going to have genes that if a gene is damaged, it typically has a backup copy, but if both genes get damaged and lead to a problem in a protein being functional, that could potentially lead to a disease state, a pathology state, and then that cell might end up dying itself or might end up acting inappropriate and causing or not completely functioning how it should. Some of the diseases that can occur, cancer is a big one that I want you to be aware of, cystic fibrosis, I mentioned cholera, but those are just a short list. So there are lots of different diseases that trace back to the cellular level, okay? And that's all I want to say about cells. You might see in the reading from Boundless, there's a little bit more on cells. And you might also see in your handout, there's a few more diseases and conditions I asked you to look at and be prepared for your quiz, your midterm, and your final exam. Okay. I am going to, what is my time? I don't even see a timer on here. I'm going to stop here and let you digest cells, let you look through cells. Look at worksheet number 3. Part of worksheet number 3 is going to review some of this material. And then I will do tissues. I'll break up tissues. I'll do epithelial tissues hopefully sometime tomorrow, maybe after I finish my Anatomy 2 lecture, and then I'll try to finish tissues, connective tissue, muscle tissue, and some pathologies on either Thursday or Friday after my doctor's appointment when I can fit it in. I know this is a little hodgepodge, but hang with me and just keep working through worksheet 2, worksheet 3, looking at the Boundless online textbook. If you have an old anatomy and physiology textbook, you can even look in there. Okay, I'm going to sign off now. If you have any questions, use the open forum or feel free to send me an email. All right, everyone, cells. Part of the week is done. We're on to tissues tomorrow, Thursday, and Friday.